Have you ever gone to the store to buy soil, potting soil, maybe for a plant or maybe to do a class project? Do we really need to go out and buy those? Are they that much better? Is any soil that you would pick up outside really just the same thing? Well, let's take some time and determine which of these may be a better purchase. I'm Roger Palmer with PASCO Scientific. This is modeling an ecosystem. We're going to take a look today at soil. Maybe you think an ecosystem we're talking about is a big area, like in the rainforest of the Amazon. Now, that ecosystem has very similar abiotic conditions. For instance, it may rain plentifully every day and be quite warm. And under those conditions, the soils will set up in a certain way that'll encourage certain types of life to flourish there. The animals that come in can live in those kinds of systems. And so you'll notice that anywhere around the world, if you go into a rainforest, they have very similar sort of aspects to them. And you may think that's the kind of ecosystem I'm talking about. Or maybe it's the Sahara Desert, which is on the other extreme. Very hot still, but very, very dry and very little life that you can visibly see in any given day. Well, I think you can find an ecosystem and some variations right under your own home, right under your own feet. Each type of soil that may occupy the grounds around your school, maybe a ripe sort of investigation for ecosystems in your own area. Obviously, it's what it's what it creates the job for a farmer to really pull them together to understand the complexities, or anybody that works in ag for that matter, to spend the time to get the most out of their own fields. So what we've done today is we're going to take a look at several soils. One you're going to see is a soil from a prairie grassland. Okay, another one that comes from a very treed area that has a lot of leaf litter underneath, and it was a pines kind of an area, lots of bark and things. And the last one is the store-bought brand of soil we talked about. And we're going to see, is there really an advantage to buying store-bought enriched soil or fertilized soil for growing materials, whether it be in your garden or in an area in a field, one soil versus another. So with that, we'll begin shortly. In order to differentiate what might be going on, how much more active one kind of soil is from another, we had to go outside and find different kinds of soils based on what was set up over those soils. That first one we talked about as being in a prairie. We went out and just dug a shovelful and made sure we broke down the clumps as small as possible. This one in the forest, again, we raised the dirt and kind of broke it up into smallest pieces possible. And took a sample that we brought back inside. And this one we bought a bag of common soil from a hardware store that had the perfect soil, had all the fertilizer and things that were inside of those. Now that we've collected those into a smaller container, we can come back to the lab and we're going to go ahead and we're going to measure out 50 grams of each of those. And so probably the easiest thing to do, and I've already done that with two of them, is to put your smaller sample that the CO2 sensor fits inside of to monitor what's going on. We're using that because the nice tight fit of the CO2 sensor is going to be able to catch whatever is in there. Is going to be in. We're going to tear the balance by taking the weight of the container off of there, and then we're going to fill the rest up to 50 grams. So I'll put this inside. Try to keep this not as messy as possible. And put that on there. There's 24 grams roughly. We can do another one like that. Maybe I'll need a little bit more, but what's nice about these soils, and even the store-bought sort of soil, I can see that there's small pieces of particulate matter that are organic in nature, maybe like a branch, bark, that have been ground fine. Sometimes they'll use just, really fine grass like moss or peat, which is short grasses that fill the store-bought soil. That's a little bit different than what I see in the forest soil. I see a lot of bark and some needles. They've been broken down quite fine as well underneath that tree area that was there. Just a little bit more. And the soil over on the far side, I can see that there's like I say, it's a little more clayed and got, the grasses as much as we could clear them out. There we go. That's a little heavy. I'll get a few more of those out. There we go. 50.4 grams. Again, about 50 grams. Now, what we're going to do is we're going to take each of these samples and we're going to put in one of the CO2 sensors. In order to do that, what you're going to be able to do is to measure all of those at the same time so you can follow the experiment and see which one's got more activity. We, again, looking for more activity in each of these soils. I'm going to start from dry so we have that as a comparison, as dry as possible. In this area, it hasn't rained for a while, so that's a good start. And then for the store-bought one, I just put it outside in the sun, and it's been about a 90-degree day, so we're hoping that that's pretty dry. I'm going to start a version of SparkView that is going to be one that I can set up. If you use the module that's with the unit, with the directions, this is really about setting up your own experiment and coming together to find out how to run a test of your own sort of imagination. And there's a lot of things you can do, especially in some kind of an ecosystem. I mean, even just with soils and obviously with the ag sort of focus that we have in these set of labs. It's the heart of what most farming occurs unless you're using aquaculture or something. But what other kinds of ways can you test besides the decomposition to see what kind of activity that's going on in there? You could be using something like conductivity sensor and pour water through to say how much salt or how much dissolvable materials are in different kinds of soils. You might be able to just use a thermometer, for instance, and determine how warm the soils get from just exposure to the sun. It doesn't have to be big or complicated. The lesson that's there has 6 different variables, like you can choose pH or conductivity, temperature. And so on our experiment that I wanted to see again is which type of soil, if any, has more value because we can see the biological activity that's going on there, which is the most active of these ecosystems that we're trying to monitor. And I thought the CO2 measure for me would have been a good experience. And so I think, if you just think of something that a little bit of data can help to support you answer that question, I think you could work on this on several ways. And so it gives you a lot of different kinds of ways to set up your own experience. It also shows that you understand, what's going on in soil that's important. The things that are dissolved in the soil, great for plants. So can you take those out, extract those, and measure them? And we have lots of different kinds of probes to do that. So as you're thinking, and it may take a little bit to design your first experience, more so than the rest of the lab even takes to measure the data, that's going to be the important part, is you coming up with the solution to how you want to measure and prove your idea, your question, is in this kind of lab write-up. It's nice because it's sort of a lab that's further on in the series. And so you should be starting to think about how you can measure your own kinds of phenomena and show that it supports an idea of what's going on inside of these soils in this example. So I just want to encourage you to think like that. That's where we really want you as a science. I mean, every field's a little different, and you're going to have to get to know those kinds of settings to be able to solve some problems. Now, one of the things you can do before you start is to make sure that you take and calibrate the sensor, which is easy to do when you set that up. And I'm going to let you look those up either with the manual that came with it, the little directions and helpful guide, or you can go to PASCO.com and you look under the videos and it'll describe that process under documents on the right-hand side in the PASCO.com for this product. So we're going to go ahead and put those down, and I just did calibrate those. We're going to measure that as well. That's the store-bought soil. Here is the forest soil. And this is the prairie soil. Just make sure they're all on and broadcasting. And you can see that the light will be blinking if it's broadcasting. I had set up to the Bluetooth beforehand to start to gather some of this data. I can double check by just clicking on here, and I see that all 3 of my CO2 sensors are ready and hooked up. And in just a minute, we'll go ahead and start to watch what CO2 is coming off of these soils because of the biological activity. All right, Now that we've got all our soils collected, we've got our CO2 sensors in the soil samples. There should be some decomposition possibly going on. Again, we've taken them right from outside. They're fairly dry. What we want to do, though, is help you to set up the graph. And like I said, I just started SparkView right from the beginning and said, let's build a new experiment. The first axes, the X and Y axes here, the X axis is going to be the time. We're going to see how CO2 develops over time. In this first one, we're going to say, let's grab, and I'll just start my way down through here. 804 is here, this one that's on the top. That is the prairie soil. So I'm going to go ahead and just click on that. That'll create one graph that's showing how much CO2 is being generated by the prairie soil. Now, under the tools area, which you can see collapse or expand here at the bottom of the interface, I can add another axis, which is great because we're going to add the second CO2 sensor in this case. I'll go over to the side and I'm going to click on that. The second one is the forest soil types. That's Y2. The third one I want to add is obviously the store-bought soil. So I'm going to add one more time. And that's going to be Y3. And we're going to choose the last CO2 concentration. Okay. Either writing these down, or if you want, you can go into making sure that you have each one of these is remembering prairie, forest, and the store-bought. That's fine, and that would be good to be able to do. I'm going to get rid of that here in a second. The other thing is you might notice is if you look at these scales, they're all measuring CO2, but they all have different numbers on the side. So those mean that the scaling is different for each one, and so they wouldn't be really directly comparable. Now, right next to adding a new instrument or a new, sensor that's going to bring some value in, it's to cause these all to be on the same scale, which is really nice. So I go ahead and click that. I can see in the bottom here all the values that they're measuring. And even if they don't start out the same because it's been sitting here for a little while we're setting this up, I am going to go ahead and start these, and we're more interested in the rate of how much production is going on. So let's go ahead and start those. We're just going to run it for about 3 minutes to make sure that we get plenty of data so that we see a trend. Sometimes right away at the beginning, it may not look like much. And as a matter of fact, you may be asking yourself, these are pretty flat. And should that be? Of course, that's a question for your analysis section, but it is encouraging to see that all three are doing the same, which is great. If nothing else, you're not quite sure why, but thinking about that as the data collection is important. If anything, Y3, which is the store-bought soil, has maybe a little trend as it's going up just a bit. Because these are very consistent, if I saw that there were, for instance, a bend in the line that was going on, I might run this for up to 3 minutes as just a suggestion, just to see if we get to a steady part of the process of delivering CO2 to the sensors. But these all seem very straightforward. So I'm just going to go to 2 minutes instead of 3 to save us just a little time as you're watching. And then we're going to go ahead and stop those, and I'll talk about what we want to get from those graphs in a little bit. We'll give it 15 more seconds. Okay, so we ran at about 2 minutes. Now, as I was saying, this is very dry soil, and even the stuff that was under the trees, which was shaded, is a little darker. You might notice as I pull this forward, you can see that the soil's in clumps. As much as we tried to break that into smallest pieces possible, that's what that looks like came from out in the prairie. No shading, just grasses around it. This one was underneath a tree, which is great. You can see it's very dark. It's got a lot of organic matter inside of that with lots of little pieces of organic matter like tree bark or needles that are decomposing. And then the last one is the store-bought. And even though we kept this out in the sun, You know, I'm not sure it was in a bag, and it was very possible that it could have either been rained on or there may have been moisture in there. This is the one that had the most of those three biological activities. Before I move on and talk about what we're going to do with the graph, though, I'm going to just do a little setup for the second step. Is we're going to try to make these all have about the same amount of water in. Obviously, we were thinking that's going to be a critical element for any process that goes on inside there, if it's a plant growing or if it's decomposition going on. We had 50 grams of soil that went inside. So I'm just going to make sure it has plenty. And we're going to put 25 mils, which is 25 grams because water's density is 1 gram per mil. And so we'll put in about 25 grams of water, just a little bit more there. We just want to make sure that we're each consistent. And we'll take this one out. That'll be good because that'll air out the carbon dioxide that's inside the sensor itself. Here, we'll go ahead and give that a good squeeze. Water's filling up pretty fast. We'll go up to 25 mils. Okay, there's the forest soil. And one last time for the prairie grasses. Another thing you might notice while this is filling up is the volume of these two that were the store-bought soil and underneath the trees is quite a bit larger than the prairie soil. So it's a much more compacted, much more dense sort of soil in the prairie material. Maybe that's because of the minerals that are there, or because it's you know, different kinds of materials, more clay or something. This has a lot more loam in it. So I'm going to put that in. Now each of these has some soil and water. I'm going to put the caps on just so I can shake them and get the water as distributed as possible through there to encourage if there's decomposition or bacteria or other kinds of things that are going on, that we can mix it as well as possible, encourage whatever, either microbes, bacteria, or even little organisms will get equal amounts of water. Okay, that's nice and mixed. And this one, let's get this guy. Okay, so we get some water on there, and I'm gonna go ahead and take the tops off. Again, I don't want to accumulate CO2. I want to see the rate. So take the tops off, put those here, and we'll keep our CO2 sensors right next to which one they started with. Now, what I want to do is to take a look, because we're still collecting data on the data itself. We're interested in what biological activity is going on each of these kinds of soils. Now, Y1, that's this prairie grasses soil, is this blue line. And if I want to see what's the rate of CO2 production, I'm going to go ahead and click once on the tool down at the bottom where there's a hand and make sure that the orange or the highlighted tool becomes the selection tool. Now, this whole line is pretty straight. I could just hit and ask for the slope of that, or if there are two segments of this line, you may notice, oh, I didn't get anything selected inside, and that's because I'm not highlighting the particular run. I'm going to highlight run 1, which is the prairie soils, then draw a box, and you'll notice now I've selected several points, and I can see that if I click once on here, I can choose the best line through here. This slope, 0.02, is very little CO2 produced every second. It's the generalized amount of CO2 being produced every second. Well, let's go on to the second type of soil. That's this one here, which was under the trees. And we're going to draw a box now that I've highlighted Y2. Notice it's got a pink box, so I'm working with the pink data or fuchsia data. Clicking that once, I can put another line through here, and the slope, I can move this along like to the bottom so I can remember which one's which. Again, is very little, 0.001. As a matter of fact, it's even got a downward trend, but we're just going to say that's 0. The last one is the soil that we bought from the store, and if I make just a quick outline there and we hit the slope, it has a slope 0.47. Okay, so where are we going to put these to store this information? In a growing table that I've created as a second page. you just go ahead and if you were making this from your own, you could create a graph and a table with that process just by hitting the plus button, and it'll ask you what kind of, here I'll show you what that looks like. I chose a graph and a table. That's the icons that it looks like, the third one down. I'll cancel because I've already created those. And the two variables I wanted to create when I go in here, when I click, are going to be user variables. One is how many hours since I put some water in each of these. I'm going to measure this every hour for another day or two to show that the rates either get faster or get slower with time. And so I'm going to the user entered kind of data. Okay, I've already created it, and that is time since we started. That's going to be the x-axis on the bottom. Okay, so that's great. I'll go ahead and collapse this again. The other one I want to put at the, and that's the time on the x-axis. Now for this one, or in the second sort of y-axis, I'm going to choose a user entered value, and that's going to be the rate of CO2 from which the slope was that we measured in the last graph. So in these cases, if we want to be to at least, let's just say 1 decimal place, because I'm not going to get too worried about this. This is 0.5, and the others are 0.0 and 0.0. So let's enter those in. So let's go ahead to the second. And in run 1, what we're going to do is enter in 0.0 or dry, and the slope for the first one is 0.0. And we're going to have to think a little bit because we're going to have to make sense of the graph when it's all said and done. And the last one, the run 3, is 0.0, and that one was 0.5. Again, I'm not going to get too carried away with all the decimals, but I do want to be able to graph these. And each hour that we measure one of these, or when you do, you just have to remember when you started. I started at 3 o'clock today, for instance. So if I get on again at 4, and then tomorrow when it's at 8, we'll just keep track of the total number of hours and what those rates are so that we can see a graph that we want to. Okay. So now we're just going to wait for an hour. And maybe if it's still in the lab, you can just mark it as even half an hour. That's fine. And if you come back to lab tomorrow, you can measure it again, and then again on the third day. Get somebody to come in after school so that you get more than just a few data points to see how these progress. That way we can enter in that data and we can look at the difference in these 3 graphs. Let's come back in a little bit once this water's soaked in and some of the biological activity has been encouraged from the water to see what the new rates are. All right, we've let the water soak into our samples here now for about an hour, and that's what I think, the nice sort of activity can start again, the biological activity that's inside of there. What I did notice in the meantime is if you look at the software, even though I had entered individual runs 1, 2, and 3 for prairie soil, forest soil, and the store-bought soil, I realized that's not going to graph out onto this graph. I'm going to need to make them in the same order. So 1 is when they were dry, and notice the 2 soils didn't give any CO2, but the store-bought one gave off a little bit of CO2 per second, parts per million per second. So I added 2 more columns. And that's always the way it is when you're designing your experiments. You're thinking, what's my best table? How am I going to get this to show up on the same graph? Because now when I plot run, each time versus each one of these runs, there'll be a different graph that we can compare. Okay, now let's go back to the previous run. That's where we left off. That was the dry materials that's on here. I'm going to hit the start button again, and that's going to start to show us what's happening to the CO2 now that we put water into those soils. And we'll give it, like I said, a couple of minutes. If it looks like it's nice and consistent for that whole time, then we can finish a little earlier, like at 1.5 minutes or something. But I think I want to let it run for up to 3 minutes, because usually if there's something that happens right away, maybe there's a little seal or something didn't happen, so you let some of the CO2 out, then it's good to sort of take the end part of the run once everything's sort of settled in. So far, they look pretty good. Okay, in this example, Y1, which is the middle green, that's sort of more beigeish green or more khaki green in the middle is the prairie soils that is here. The blue, Y2, okay, is from the forest soils. And then Y3 is the store-bought soils. Now, don't be confused about the height of those, but it's the rate at which the CO2 is being produced when you're thinking about this data as it's coming out. Okay, they look like they're pretty consistent. I'm going to go for 1.5 minutes. I don't see any changes. If you need to, I would say as long as you run out to 3 minutes, if you sort of notice that the slope's going this way and then changes a different direction, it gets more steeper, a little shallower, run it until you got a nice consistent set because it's the slope we're interested in this one. So there's about 130. I'll go ahead and stop. All right, now I'm going to go back up to run 2. I'll click on that first example, which is this middle value. It's the lighter green of the two, okay? And you can tell because if I've got this highlighted and I draw my box, I make sure that the box is highlighted. It looks like you can see the box. It will show up as selected, okay? And it did. So I was clicking the right one. I'm going to put a slope on here and I'm going to drag the box off so I keep it close to the line, and that way when I'm flipping to the next page, you'll say, oh yeah, that's the one that goes with this line. Okay, now I'm going to move over to the blue line. I'm going to draw another box. This one, I'm going to try to just get it so that we get as much of the end of that line as possible. We're going to hit the slope tool, and I can move this slope kind of out of the way so that we see it's part of the blue line. The last one, I'm going to switch to the more of the Kelly green, and I'll make a box here, and we will get our best line through that, and I will move that so it's up here and we don't confuse which value stands for which one. So I'm going to click on the first one again, Y1 in our cases, and I can double check by looking at the number on the sensor, especially when you're using the same sensor for all 3 of these. It can be a little confusing. But I'm going to go look here, and it says, the one that ends in 804, which is the prairie soil. Okay, I'm going to add that to the prairie soil one in the next box, which the value here is 2.58. If you want to just go to the 10th, that's fine. The prairie soil, let's store forest prairie. Okay, I'm going to go ahead and touch that. 2.58, if I told you I was going to just stay to the 6th, to the 10th. So that just makes it easier. And the time now we've run is 1 hour. Come on, there we go. We'll put 1 in there. Now let's go see what the forest soil was. And that is this second one in blue. And double check again. The blue Y2 says it is 536. And that is the number on the soil for the forest soil, and that has a 1.19 or 1.2 slope. So let's put that in as a value. 1.2, and that's parts per million per second of CO2 generation. That's how much CO2 is being generated. Bigger number means more biological activity in the soil. The last one, we're going to go down and I'm going to highlight the third one just so you can see. Again, this is just to double check to make sure. That is 721, that's Y3, and that is 721 in the graph. So it's this top one, and that has a 2.58 for the store-bought soil. And so I'm going to switch over to my second graph, and I'm going to put in 2.58 for the rate, or 2.6. Again, I think that'll be just fine in terms of our comparisons. So with that in mind, every hour I'm going to take, if I can get a chance to come back between classes and just hit the start button, I can get another run. All these runs are being saved. And then you can do the analysis. If you can get a couple in today, then come back tomorrow and get 1 or 2 in. By the third day, you should have a good number of points on your line. And if you're using this from home, and this is maybe a day when you can't be at school, then use my data set and you can do the calculations here to fill the rest of them out. All right, we'll see you in another hour. All right, so we've been able to collect data now for 2 days. and see how much of the biological activity is going on in each of these soils. One from a prairie grasslands area, one from a forested area, and then one from store-bought soil, like a vegetable garden soil. It's been fun to see, and as the results are coming in, and we've got some interesting things that we can explain the graph of how that rate is going on. But we're going to take one more measurement to get the last field here. And what we're doing is we've connected we still have connected. And in between each session, because it's been long enough, like overnight, for instance, we've reconnected these. I just made sure I paid attention to which order they came in to make sure that we were getting the data from each of the soil types. And that's something you'll have to think about when you're designing your own experiment of making sure, each of these instruments is giving you the data that you'd want. But let's go ahead and run this for another minute. And we'll get our last run. You can see one typically has a lot more carbon dioxide in it. And that may be due to the rate, even though we took each of these and we made sure we had calibrated it in its own clean sample jar. Because the carbon dioxide rate is about 400 parts per million for the CO2. It's described well on the website, PASCO.com, under the documents section of how to do that. And we'll just make sure that we get a good run for about a minute. Seems like a minute's well, unless there's still some, like it might have a little bit of some air can get in through the side or something would happen. And so, in which case I'd like to get at least a minute of good, looks like steady data that's coming off of each of these. Okay, and there's our minute, so I'm going to go ahead and stop. We're going to use the same tools that we have been for each of these to measure that, and that is, first of all, the selection. I can tell that my selection is good here on SparkView because it's the square that's highlighted. I'm going to start from the first set, the green set, and that's the prairie soils here at the bottom. And I am going to just stretch a square over that data, and then we'll hit the best fit line, or the linear fit. I'm looking for the slope here. That's telling me the number of parts per million CO2 change per second. And then we're going to go ahead and switch that over to then the forest soil. That's next. And then I'll draw a box. Notice the color is going to match the line color as well. We'll do a line fit. That's great. I'll put that next to it. Just again, putting these data next to the what they're measuring is always nice, so you don't forget, or especially when I have to change the page. And then I'm going to go for the last one, which is highlight that third run, Y3, and I will run a box over that collection. And we'll get our line of best fit, make sure that's the highest one. All right, so we're going to take those numbers and we're going to transfer them to the table. We're going to start with the soils that came from the prairie, which is 2.25. That's the rate of CO2 production, 2.25. And then let's go back to the second one, which is the forest type soil. And that had a rate of 0.90. I was reading the wrong thing here on the bottom. I can see it's 0.843, a little lower rate. So let's go change that again. 0.843, 0.84, and then let's now go to the forest collection, which is 0.97, 0.907, so 0.91. Back over here, 0.91. And the last one, which is the store purchase soil, and that is 1.65. 1.65. Okay, so we're 47 hours later, 2 days later, and I'll go ahead and hit that enter. We can see that we've got this graph of activity that's going on. The soil, the third type of soil, the soil that is the store-bought soil, seems to be this top line, and then sort of both sometimes one being over the other. Both the forest soil and the prairie soils are right next to each other. So that's the data collection, and those are the rates of reactions that are going on, which is important. We're saying that is the key measure of the soil health, and what's making nutrients available to the materials, the plants that are growing in each of those soils, because the decomposition is providing the nutrients for whatever it is that you want to grow there. So we're using that as our measurement of soil productivity. Now that you've finished collecting your data for these experiments and whatever you've done for those experiments that you have possibly done, make sure that you've collected all of those. You've turned now the runs to be showing that we have. We've also gone and expanded in the bottom of the tools. We've expanded the data so it fills the screen. I've gone through and labeled each of those graphs, because sometimes it'll be confusing to say, which type, especially if you're using the same instruments in 3 different conditions. Again, notice in terms of setting this up, we've only changed the place of where the soil came from. So we're being careful to choose to vary really only one condition. We use the same mass of soil in all those cases, and we put the same amount of water in early on at the beginning, so that we were controlling as many of the variables as possible. For you now that are ready to turn this in, now that you've got your graph, taking up the largest area of the space in the software, go ahead and label those. I can use the little text tool that's at the bottom of the software. You will need to make sure that the run, like Y1 versus Y2 versus Y3 is highlighted. And then you can tell me which is the store-bought soil, the forest soil, the prairie soil in my example, or whichever cases you have as you set up for your experiment. Now, as we look at this, we're taking a look not only at just which is a higher, and these are rates, and so like we said, it's the biological activity going on in the soil, but not just in a direct order, but you have numbers to really compare these, saying it's 2 or 3 times more active. It's fun in this case that we saw as those the reactions, or as the sort of soil has been sitting there, we were hoping to see what was going on. Was there a chance for more organisms to start growing? If it's microbes or something like molds or funguses or decomposers, is there evidence that there's more of them coming into the scene? So you could be talking a little bit about that activity is increasing. Why? Why are there more decomposers, or why is there more decomposition rate going on? And you have to be careful when you say these in your lab write-up to include, you think maybe there is more bacterial growth or fungal growth. And then at some point it looks like there's a rate that's going down, and could that be explained? If there is a case like in the bottom where between the forest soil here and the soil that came from the prairie, that there seems to be a trend. There's generally lower activity in one of these, except for one time. Just explain that in your conclusions, and go through and rank order these for my experiment. I want to rank order which is the lowest through which is the highest. And even though there's a little bit of exception, we can then give a short explanation of why we still think, there's still one that's lower, even though there's one data point that's outside that for most of these that we ran. I can go back to that run, for instance, and say it was a noisy run, or there was some data that comes in there. But those are important things to be talking about when you're talking about your experiment. So even though you make some conclusions, people can look back at your data and start to make some realizations. One thing you might notice here is that there's a large space in between the bottom of the soil and where the detector is. So that may have made a consistent difference as well in terms of what the CO2 is, because the CO2 has to be generated and it's going to fill this before it starts to be registered inside of the detector. When you're all done, you've written up a short description. You can take a screenshot and include those, but then turn that back to your instructor with a great write-up of saying, here's your question, here's the procedure that we've done, here's the data that we've measured, and then, like we've been doing right in this last part, we talked about some of the things in the analysis that you want to include. including the shape of the graph, at times when it's going up, what's going on there, why it's going back down now 2 days later. As we ran this experiment, we took these out and then turned them off, so we didn't run the battery out. So there was evaporation going on as well. So those might be some great ways to include variables that might be changing. And that will be a way to show that you understand the full science process. When you finish, a nice summary line of saying my top soils, or was it worth it to buy garden, provided commercial soil? And you can say yes or no, and here's 2 or 3 of the reasons. You may feel like you're just repeating yourself from the previous point, but making a good, solid last statement to conclude based on your original question is going to be the important thing for when we talk about modeling an ecosystem, and for you to get a good grade when you turn this back into your teacher. So good luck with these, and whatever your experiment was, you can use this set of data that's there as the provided sample data, or you can use the starting experiment, Spark Lab experiment to start your own experiment. But remember those parts, because they're still going to make it give you the best sort of response from your teacher. Thanks, and good luck with your lab.